All right. We're ready to get started in our afternoon session. We have Sandrine Bedard, is a biomedical engineering and current PhD candidate at Polytechnique Montreal, training with Dr. Julian Cohen-Haddad at NeuroPoly. Sandrine's research includes spinal cord image processing, developing new methods for the spinal cord toolbox, and improving spinal cord functional MRI. Sandrine is a rising leader in spinal cord imaging, leading several highly impactful projects, including the contrast-agnostic spinal cord segmentation model and the new spinal rootlet-based registration method. So excited to have you here, Sandrine. You can take over.
Great. Thanks for the introduction, Ken. So first, I'm going to start by presenting a bit myself, maybe a bit differently than how Ken presented myself. So I'm a PhD candidate at Polytechnique, and I'm currently co-supervised by Ken and with Julien. And I actually started working with image analysis back in 2020. So I'm going to take you back. In 2020, it was COVID. I was a third-year undergrad student in engineering, so this was me at home. And we needed to find an individual project to do. So I contacted a prof at my school. So I contacted Julien, and I asked him if he had a cool project for me. And he suggested this project to me. He was like, oh, we have this huge data set, the UK Biobank. You could use it to measure the spinal cord upper cross-sectional area from brain imaging. And also, he recently did the Spine Generic project that now you all know about. So you can follow the steps of the processing pipeline and apply that. So I opened my terminal after I figured out how to install the spinal cord toolbox. I tried to segment the spinal cord. But, well, it failed. So this was the image I had. So then I went back to Julien, was like, oh, it failed. Then he asked me to open an issue on GitHub, of course. And then he said, well, there's no spinal cord. So it was actually brain extracted. So that's why the segmentation model failed. So I went back to my computer. Then after getting the raw images, I used SCT again, retried to segment the spinal cord. Well, it failed again. So this was what my image looked like. So I went back to Julien, opened another issue on GitHub, and then he told me, oh, gradient nonlinearity correction was not applied here. So like if I didn't know where the spinal cord was in an MRI, like gradient nonlinearity, I had no clue about this. And finally, I find a software. I find the files to apply this correction, and I was finally able to segment the spinal cord. So a few takeaways from this was my first experience with image analysis of the spinal cord. Well, it was not really like following the recipe of Spine Generic. It did not work directly here because you need to look at your data and also adapt with your goal. Here I had brain images, so it was a bit different than what we had from the Spine Generic protocol. So I'll take you back now to today. I can reassure you, I know now how to analyze spinal cord images. I know where the spinal cord is. And today I'm going to mainly focus on structural analysis of the spinal cord. So first, I'm going to talk about spinal cord shape analysis. Then continue a bit about template-based analysis. We're going to analyze also the shape of other regions within the spinal cord, like gray matter lesions. Then talk a bit about pipelines and workflows for analysis, and also finish with some new upcoming features for spinal cord image analysis. So let's start with spinal cord shape analysis. So the first step that you will probably do is the spinal cord segmentation. So you can take one of your beautiful anatomical image and segment your spinal cord. And with that segmentation, you can extract metrics of the structure. It can be used to align participants to a template space, or it could also be used to co-register some contrasts together. But one problem that we currently have with these segmentation methods is they actually depend on the MRI contrast. So for example, if we segment a T1 weighted image and the T2 weighted image of the same participant with any segmentation model, then these are the segmentation you would get. And if you measure the cross-sectional area, well, you would get different values. And this is problematic because, yes, these are two different contrasts, but even if we have just various sequences and parameters, this can be limiting for large multi-scale studies. So our idea in this project, so this was the contrast agnostic segmentation, was since we know our segmentations differ within one participant, we would like to just create one unique segmentation per participant. So first, we used the Spine generic multi-subject dataset that had 267 participants with 6 different contrasts. Then we segmented all the 6 contrasts individually, and we aligned each contrast to the T2 weighted contrast. So here, then, we brought also the segmentations to the T2 weighted space, and we computed a weighted average. So at the end, what we had was a kind of probabilistic segmentation, so with values between 0 and 1, that would kind of consider the boundary of the different contrasts here. Then we brought back this unique segmentation to all the individual image space just to get back to their original resolution. And with this, we trained a new deep learning segmentation model, but with these segmentation instead of the ones that we had before. And with this, then we measured spinal cord cross-sectional area average at C2, C3. And with the previous method, we can see here that the CSA varied quite a lot across the contrast because these are same participants. But now with our new model, we were really able to achieve stable cross-sectional area across these 6 different contrasts. And we're still working on improving adding other contrasts to the model and also monitoring to making sure that this CSA stability stays even if the model changes. So another important step of image analysis is vertebral labeling. So usually we want to label the intervertebral discs. For example, can be done automatically with total spine seg, a method developed by Nathan, a PhD student in my lab. And it's useful, for example, if you want to extract metrics at specific level, for example, in cross-sectional area, or to get lesion location, for example. And it can also be used as an anatomical landmark for template registration. So now that we have our segmentation, we have our disc labeling, an important step is always quality control. You need to look at the results of your segmentation and your disc. So what do you look for in this? First, for segmentation, you can look usually for maybe under segmentation. This can happen sometime if there is a lesion. So this is nice. Then there can also be some leaking outside of the spinal cord. And this is an example of a good spinal cord segmentation. Now for disc labels, what you can look for, we can see here the C2, C3 disc label. We see that it does not align with the discs. And same thing here. And on the right, this would be a good vertebral labeling. So all of these images, you can actually get this in this nice report that is done in the Spinal Cord Toolbox. So on the top, you would have your image, a mosaic of your image. You would have the command that you ran here. And you can actually put these check, so if it's good, if it's bad, for all your images that you processed. And then you can download this list, so you have a list of what segmentations to correct. So now we have our segmentation, we have our disc labels. One thing we can do is compute cross-sectional area. So this is an example. We could average CSA across C2, C3 vertebral level. However, we know that the location of the vertebrae and the spinal levels is not a perfect correspondence, and it also suffers from intersubject variability and also with the positioning of the neck. So another way that you can compute cross-sectional area is instead to base it on another landmark. So here we suggest to use the pontomedullary junction. And it's quite useful since it's just one unique label that you need. You don't need the multiple labels like for the discs. And it would be computed at a distance from the pontomedullary junction. So now here's an application of what you can do with cross-sectional area. So usually in MS, CSA is a measure of atrophy of the spinal cord. And here, automatic segmentation of the spinal cord was applied to this large MS cohort. And it was also compared with some semi-automatic method. And it was shown that the CSA did not differ if the automatic method versus the semi-automatic method was used. And having a fully automatic method really ensures that you remove user bias, and it also makes it easier if you want to analyze large database in a fast, reproducible way. So another thing now we have are segmentations. We can also measure CSA on healthy individuals. So this is an example of cross-sectional area across all the cervical spinal cord on 203 healthy individuals. These were the spine generic data set. And we can clearly see, for example, the cervical enlargement. And across the subject, we had about an 8% inter-subject variability. Now, if we look also at the CSA curves, but now across the entire spinal cord, if we look in the lumbar region, we can see the lumbar enlargement, and we see that its location varies quite a lot in reference to the vertebral levels. And also the location of the tip of the cord, even if we say it's around L1, we can see that this is among 50 subjects. It varies quite a lot. So these are important things to remember when you want to do some comparisons across subjects. So let's dive a little bit more into what are the sources of the variability. We also started a bit in the questions in the previous talk about this. So as Julien mentioned, the MRI vendor, the sequence parameters, if there are some filters, this affects the CSA values that you have. Then also, male have a larger spinal cord than female, so this is also something to note. Age also can have an effect on CSA. So it was shown there was a trend that CSA increases until the age of 45 and then decreases. But it's still a bit unclear what is the effect of age on CSA on normal changes. Because, yeah, these fits were not significant in this study. There's also a correlation between cross-sectional area and the height of the person. And this correlation is even stronger if you take just the cross-sectional area but of the white matter. We'd find this same kind of trend also with weight, but are a bit weaker than for the height. Then the size of the spinal canal is also correlated to the size of the spinal cord. And brain volume is also correlated with the size of your spinal cord. So now we talk a lot about cross-sectional area, but there are other different shape metrics that you can actually do with segmentation. So, for example, you could measure the anterior-posterior diameter. There's the right-left diameter. Other a bit more complicated metrics, eccentricity, solidity, or orientation of the spinal cord. And these are particularly interesting if you want to study, for example, spinal cord compression, like in degenerative cervical myelopathy. So here on the left panel, we show an example of a mild compression. And on the red dashed line, we can see the corresponding metric of this compression, which would help identify it. But the thing is, spinal cord compression, there's so many changes that occur. It's hard to have just one metric being to help you really characterize it. So we recently tried to add some symmetry metrics of the spinal cord. And one of the hard tasks with this is to find the axis of the spinal cord, which is the first step to kind of get this symmetry metric. And then what we did, so here is an example slice. We flipped half of the spinal cord, and then we compute a die-score. So this is the overlap between the two of them. So here, for example, the blue curve with the blue arrow, we see that this corresponds to this slice where the spinal cord is very asymmetric. So now that we have all these metrics, and we know that there are some intersubject variability, how do we compare across individuals? So yes, like I told you, you can, for example, average it at C2, C3 vertebral levels. But here we have some beautiful isotropic image, so it would be nice if we can have some slice-wise metrics. But how can we put them in the same space? So here we worked on a normalization technique that does not include any registration. So these are transformations that are applied directly, like on the CSA measures, and we do some linear interpolation to put them in the anatomical space of the PAM50 template. So this is directly applied like to your CSV file, because we know that like registration would maybe deform the spinal cord, and we really want to measure the metrics in the native space. So now with this, what you can do, and you can have these very nice slice-wise plot that are a bit more specific than if you have something that is average across an entire level. And we also know that a spinal cord compression may not cover an entire level. They're mainly more localized, so it helps capturing more subtle changes. And this is, for example, a spinal cord compression. We measured the anterior-posterior diameter. This is the red plot that we overlaid on normative values of healthy individuals, and we can really see the drop of AP diameter at C5, C6, which corresponds to the compression. So this is another example on an application on a cohort with degenerative cervical myelopathy. This was a study led by Fazia Muhammad and Zach Smith, and this was with age and sex matched cohorts. And we can really see that the cross-sectional area is lower in our patients, and it's also more pronounced at the intervertebral discs location. So the thing is, you don't always have some age and sex matched cohort. So how do you get these inference at the individual level beyond group averages? So this was a project led by Merve Kaptan, which is still ongoing, and it's to do normative modeling. So the concept here is we created a model on a large database of healthy individuals, and we included the cross-sectional area, but also sex, age, height, weight, and the vendor. And with this, we create a kind of z-scores to know how much you differ from the normal population. So here in blue, we have some healthy individuals. We see that their z-score is mainly around 0. And in red, these are some DCM patients, and we see that they have a drop of the z-score compared to the healthy population. And if we look at some individual plots, so for example, this is a subject that has a kind of generalized atrophy of the spinal cord. But here we could see this would be a patient that has maybe more localized sites of compressions that we can really see here with the z-score. So another way to quantify the spinal cord compression, you can compute the MSCC. So this is the maximum spinal compression. And this uses healthy levels above and below to kind of self-normalize the metrics. So you will also be able to compare with other individuals. But one problem with using directly this metric is that we know that the shape of the spinal cord varies in the superior and inferior axis. So for example, if you have a compression at the cervical enlargement, and then you divide with the level above and below, you would probably underestimate the severity of this compression. So one way to mitigate this is to add this extra term of normalization that uses the metrics computed at the same location, but now in a database of healthy controls. And for example, when you have this, you could correlate these measures of MSCC with some clinical scores. So this is the same study that I showed before, and here they correlated MSCC of a spinal cross-sectional area with some dexterity scores. And we see, so the higher the severity, the MSCC, the lower were the dexterity scores here. So far we have mainly concentrated on spinal cord, but you can also segment the spinal canal. So recently, an intern in our lab, Abel, worked on training a deep learning segmentation model of the canal that works on T2 weighted images. It's also robust to different orientations, so it works on axial acquisition or sagittal, and also for cervical, thoracic, and lumbar spine, and different pathologies. And with the spinal canal, you could compute a ratio of the spinal cord to spinal canal. So this is the division of your spinal cord cross-sectional area to the canal. Sometimes this is also called spinal cord reserves or the spinal cord parenchymal fraction. It depends on the field. If you're more in DCM or like in MS, sometimes it's called differently. And here in blue, I overlaid the A score of this patient. And we see that there's an increase of this occupation ratio at that corresponds to the location of the compression. So now let's continue with template-based analysis. So Julien quickly introduced the PAM50 template is the standard for spinal cord. It was PAM stands for Polytechnique, Aix-Marseille, and the Montreal Neurological Institute that collaborated on this template. So it includes T1 weighted image, T2 weighted, T2 star weighted. You also have probabilistic atlas of the gray matter, of the white matter, and you also have a white matter atlas of 30 different tracks, and also the gray matter parcellation. So how to register your images to the PAM50 template. So first, you'll take your previously segmented image and your vertebral labels that you already have. Then a straightening of the spinal cord is aligned because the curvature can vary across individuals. And there is an affine transformation that will align the vertebral labeling, so your anatomical landmark, to the ones of the PAM50. And then there will be a non-rigid transformation that is done in the axial plane just to align the size of the spinal cord. And then all these warping fields are concatenated to each other, and you can get your back and forth transformation. So you can bring the entire template to your native space. And for example, you can do some atlas-based analysis. You can compute, for example, MTR values in specific tracks that you would want. So there are different parameters that you can change in this registration pipeline. It's quite customizable. And I'm going to focus mainly on the anatomical reference, so the vertebral labeling. So as we know, spinal levels are not equivalent to vertebral level. So the spinal level is defined with the entry of the nerve rootlets. We can see this is the correspondence in a T2 weighted image. And even if there is a correspondence between spinal and vertebral bodies, well, this varies across individual. And it's also more pronounced when you go to the lumbar level, for example. So one thing you can do if you're interested in lumbar imaging is you can use other landmarks that are not related to the vertebrae, like the location of the conus medullaris, for example. Another thing you could use is rootlet segmentation instead. So recently, Jan Valosek and Katerina, they developed a segmentation model for nerve rootlets that works on not only T2 weighted image, but also MP2 range images that segments ventral and dorsal rootlets from levels C2 to T1. And what you can do with this is you can use them in your registration. So this is an example of the anatomical images of 226 people. And when we use the rootlets, we can really see a good alignment. We can see the delineation of the rootlets, while when we register with the disk, it's kind of a blur. And I think this is even more pronounced when we look at the ventral rootlets, so we can really see the shape of the rootlets across the participants. But when we align with the disk, it's really a blur, so the levels are not really aligned. And an interesting application to this is really for fMRI. So we tested this method on fMRI of the spinal cord with an electrostimulation aimed at the C7 dermatome. And we can really see that we get a more localized activation and higher z-score compared that to the disk. So let's take a look at some shape analysis of other regions of the spinal cord now. So first, the gray matter. You can segment the gray matter automatically. We have Nilser, here a PhD student from my lab that worked on a deep learning-based contrast and region-agnostic segmentation model for gray matter. And it works across 3 different fields. Field strength was trained on 12 different contrasts and a lot of various pathologies, and also on all 3 regions. And one thing you can do when you have your gray matter segmentation, here this is an example that it was applied on patients with amyotrophic lateral sclerosis. And it was demonstrated that the spinal cord gray matter was actually a better biomarker than spinal cord atrophy directly. And it was also better for distinguishing patient from healthy controls for the 6-month prognosis and motor score. Now, if we also take a look at lesions, so you can segment lesions from multiple sclerosis patients. This is work from Pierre-Louis, a PhD student from the lab, that worked on this deep learning method that is robust to a lot of contrast, image orientation, MR vendors, and field strength. And an example of thing that you could do with that is you can take your lesion masks from your images, and you can register them to the PAM50 template, and you could create a lesion probability map of the location of the lesions. We also have in a Spinal Cord Toolbox a model to segment lesions from traumatic spinal cord injury. This was work from Jan, a postdoc in my lab, and Naga. And they worked on this deep learning segmentation model for intramedullary lesions on T2 weighted image. And this model works on various orientation, resolution, field of views, and also on traumatic and non-traumatic spinal cord injury. And with these segmentation masks, you can calculate, for example, the number of lesions, you can calculate the lesion length, and also, for example, tissue bridges. So now I'll talk a little bit about pipeline and workflows. So I'll show you an example of the type of workflow that I use when I analyze spinal cord images and a way to make it reproducible. So first, data organization is very, very important. We follow the BIDS convention, which is a standard for neuroimaging data. And then you will run an automatic pipeline on your data that will perform all the steps, segmentation, disk labeling, and ideally your code will be on GitHub, also version tracked, and you would run it on all your participants, ideally without any manual intervention during the pipeline. So you can go to sleep, go check on the next day, and you'll have your results. Then you can continue, do your quality control, do some manual corrections if needed, and you can add your corrected data to your BIDS data set inside the derivatives folder. So we have, for example, spinal cord segmentation, disk labels, and then when you rerun your analysis, it will not run the automatic method if it finds the file that is in your derivatives. So that way, if you rerun your pipeline, you won't need to redo the manual corrections, and this ensures that your pipeline is reproducible, and then you can get your final results on your entire cohort. And having good data organization, fully automatic pipeline, it will make you more confident when, for example, reviewer 2 asks you to rerun your entire pipeline and you're very scared that you won't get the same thing or it will just crash. This makes you more confident that this will not happen. So now I'll actually pass the torch to Julien, who wanted to talk to you about cool new features that will arrive in SCT very soon.
Thank you, Sandrine. Hello again. So some of the new features, because we have others, but one is the for many years the labeling of the vertebrae and the disc was not working very robustly. It was failing approximately 20% of the case across multiple data sets. So then Nathan Molinier, with a PhD student, worked on a version. So it's very similar to the total seg model that some of you might have seen for the head and the body based on CT scan, but this one is for MRI, and it's particularly designed for the spine, because what total spine seg does not do is provide semantic labels of each vertebrae, and so that's what total spine seg does here. And it's robust to multiple contrasts, field of view, resolution, and so on. And then one thing that actually we work with Ken and Dario and other collaborators is a way to get some quantitative metrics from the discs and the vertebrae, particularly for lumbar diseases. And then generate reports based, for example, spinal canal size, or the health of individual intervertebral discs that could then be compared to a normative data set, and then check whether the disc is healthy or not. And on a similar basis, I was talking this morning about this Kaggle RSNA challenge, and so that's was the work of Thomas Dagoneau, who developed a model that uses total spine seg for the identification of the patch where each intervertebral disc is located, and then that patch is analyzing around each intervertebral disc to basically classify or get the probability of 3 different diseases: neural foraminal narrowing, spinal canal stenosis, and subarticular stenosis. And that outputs a probability for each of the intervertebral discs. And then there's also a student who contacted me, so that's a bit convoluted, but there's a link to Stanford, because he's actually a student of a researcher in Thailand who knows Kawin Satsongpop, who knows me. So that's how basically that student ended up in my lab. And he's working on a new method to correct for motion in diffusion and fMRI time series, because there is still room for improvement. And I don't know if Stefan is here, but yes, excellent. So and also Alex, yes. Okay, so that's actually the work of Alex. What you see on the left is SCT that runs automatically on the Siemens console. So now it's possible you get an anatomical scan, and then a couple of seconds later you get the segmentation as well. It is now possible, and it's possible thanks to what's called Open Recon ecosystem that Siemens provides. Previously, it was a whip called Fire, F-I-R-E. They like to play with fire and ice, but this one is Fire, and then you can run any Python code that is embedded into Docker container. And that's also the strategy that Neurodesk has been taking by exposing a lot of tools from the neuroimaging community within the Siemens console. So I strongly encourage you to look more into that, and I think that it really opens up a variety of possibilities, exciting possibilities. There you go.
Thanks, Julien. So I wanted to end with a few take-home messages. So I showed you a lot of different things you can do to analyze your structural data. So there's no recipe to know what to do. You need to decide how your data looks, and look at your data, look at your results, and the more you script also, the more reproducible it will be. And if you want to know more about exactly how these tools work, we have a workshop on the Spinal Cord Toolbox that is in December, free and online, and we do some hands-on examples of this if you want to join. And I also want to thank everyone at Neuropoly and also at the Stanford team, and thank you.
All right, thank you, Sandrine. We're going to open it up to everybody here first. Any questions for Sandrine? Go ahead.
Thank you so much for the talk. So this question might be a little bit outside the scope of your talk, so I apologize if it is. But like when you're doing, like, say, diffusion-weighted imaging, those additional pre-processing steps, like maybe the distortion correction or eddy current correction and denoising and stuff like that, are they done in this toolbox, or how does that kind of work?
So for diffusion imaging, you would do the basic steps were a bit like Julien presented from the spine generic. So basically more motion correction, and that will use like the segmentation and registration to the template to bring back your atlases. I don't think in the spinal cord we do much corrections for eddy current or for distortion either. But yeah, I don't know if some studies have tested, but it's not necessarily done, I think, as it is done for the brain.
Cool. Thank you.
I can maybe add
Yeah.
to that. Yeah, so it's a good question, and so there's no distortion in the spinal cord. No, I'm joking. There's a lot. So about eddy currents. Eddy current, as you probably know, happens as the further away, because there are shearing transformations. In addition to the translation are taken care of by the motion correction, and the shearing is a linear function of where you are from the isocenter. If you're exactly in the isocenter, you don't have any shearing. And the thing is, when we do acquisition of the spinal cord, usually with diffusion fMRI, we place it along the isocenter. So there is not so much XY shearing in that case, because that would be only in plane, that would not be through Z. So that's, part of it could reasonably be taken care of by the motion correction. About distortion, there is an ongoing debate about applying top up or like blip up, blip down, field map based, and so on. It's not obvious or that it's generally a good idea to do that. I've seen cases where it actually brings more problematic, it actually introduces distortions, because having a reliable field map in the spinal cord is difficult. You get noise, you get also, because of the bony structure doesn't bring so much signal, you get, like unreliable field maps, and that could actually do more harm than good. And so one of the strategies to actually, as Sandrine said, registering to the PAM50 using nonlinear registration, or registering to the anatomical T2 weighted image. And so if you do that with nonlinear registration, that somewhat also address the problem of distortions. And there's a long thread, discussion thread on the forum about that particular question that Saj Babdi from Oxford was actually raising.
Great question. And there's also similar sort of unknowns regarding functional MRI, and could you use these distortion correction techniques for functional spinal cord imaging? We don't know. We don't employ them, but some people do. Any other questions?
Thank you. This is a question about a CT. So currently trying to run it on a Mac. It requires Rosetta emulation. Is there any like showstoppers or blockers that not allow a CT to run on the Mac processors?
Just issues with installing SCT on the Mac? Is that right?
Yes, so it
Yeah.
requires emulation between the processors still. Is there?
It should not.
Well?
No.
Okay, I'll talk to you later.
Yeah, and then
We can look at it together after that.
yeah, after that, bring it to the pizza party. We can do it there, and then also tomorrow in the hands-on session. So if anybody needs help installing software, trying things out, there'll be lots of time for that.
Hi, I have a question related to what you showed about the cross-sectional area, not exactly with the cross-sectional area. What has been your observation about when you see there is some difference between the subjects in the cross-section? What has been your observation regarding the longitudinal difference? I mean, along the z-axis, is that stable across subjects, or how much because it affects the slice thickness and things like that. So have you ever looked into that part?
So I think here are these metrics that I show were computed on images with 0.8 image, so millimeter isotropic resolution. So so it's quite high. So this ensures that we can get some good, I think, like stability across time. But of course, the thing is always like, if you want to compare two images and your participants are not positioned in the same way, then maybe yes, you would use vertebral levels, for example, to do this alignment. But we also showed that even if with different positioning, this can change. So it's always this kind of difficulty of having a good landmark to do some longitudinal changes. Yeah, longitudinal stability. Also, just ensuring, for example, that you use the same sequence, the same contrast, because this can also affect, even if you use the new segmentation model. These are also things that would affect your measures. So always try to minimize those kind of variables. But usually we can get the intra-subject variability would be under like 4% for different sessions, if there's no pathological changes.
Okay, yeah, thanks.
Was some of that about the termination of the spinal cord itself and the variability?
Right.
Yeah. Did you have a question, Suzanne, or you have an answer to this?
No, I don't have an answer.
Oh, let me go up there then. So kind of the variability like between T9, T10, L1, L2, and I think that there just is a lot of variability, and kind of like, you know, like a classic is like the lumbar puncture example where, I don't do them, so, but L4, L5, you go way down this way so that you're not going to hit the spinal cord. But there is this much variability. There's quite a bit of variability on where the cord terminates with respect to the vertebral level.
And also between the segments, between C3 versus C3, C4 versus C4, C5. Are these comparable in their dimensions?
Oh.
So there is, usually each spinal level will have probably different lengths, but also like if you go from a subject to another, it's not just like a linear stretch of the cord. Sometimes some people will have like longer cervical segments than another person, but they have the same length of the cord. So there is also quite some variability in the length of each segment, and just within a subject and across the subjects.
And then this is calculated without, over here is cervical only, right? Without vertebral-based normalization. And you can see the enlargement for sure. When you do it with the rootlet-based registration, it looks a similar curve, right?
Yes, so there is less variability between, like, at the cervical level between vertebrae and spinal level. So that's why we can still quite see well like the different shape of the cord. But when you look like at the lumbar, when you average, for example, this curve at the bottom, their average, we see that it kind of flattens this lumbar enlargement because of this variability. But yes, I think you could also do these measurements in reference to the rootlets instead, for example.
And then the rootlet-based registration technique is limited to C8, or does it go to T1? T1. So it's, still in development.
Yeah, segmenting. So yeah, nerve rootlets are really, really small. If you think the cord is small, nerve rootlets are even smaller. If there are artifacts in your images, like for example CSF flow, or even with compression, it makes it harder to visualize them. And also, so when you're at the cervical level, the rootlets are quite parallel to the cord, which makes it easier. But the more you go down, the more they're angled, and they really like follow the cord. So it's very difficult to identify them and to follow them back up to a spinal level. So that's why like having automatic methods to do this is challenging, and it's still ongoing work to try to get it down, maybe someday to the sacral, but for now we're just at T1.
So I'd had a sort of less interesting question with regard to what you were just discussing. If you measure the location of the conus with regard to either the rootlet method, which you've already pre-answered now, you can't do that in the lumbar spine, but have you tried measuring the distance of the conus from the pontomedullary junction, and is the variability the same? Because as Ken points out, in actual patients, there's tremendous variability, but I was wondering if that got smoothened a little if you measured from the PMJ.
So actually, I did some test on ex vivo spinal cord to compare the location of the cervical segments, but with the length of the spinal cord, so the distance, I think it was from the C2, the beginning of C2 spinal level to the conus medullaris, and that did not help.
Okay.
Because the thing is, it was really that it's not linear. It's not like a beautiful, you can divide by the length and then you get a beautiful curve that it's perfect for everyone. And even, we could not predict the location of each segment. Each segment had different lengths across the participants. Even that, we're not necessarily able.
I had a second separate question. You showed a beautiful slide where you had defined the location of spinocerebellar, spinothalamic, corticospinal tracts, cuneate and gracilis.
Yes.
Do you assign those locations based purely on geography, or in the different contrasts, are there imaging characteristics that help you define those tracts?
Yes. So the only characteristics that is used is really the shape of the spinal cord. For example, if you register using a T1 weighted or T2 weighted, it would be the shape of the cord. But you could also help with if you have a gray matter segmentation. So you could use, for example, the white matter mask to improve your registration to the template. But this is kind of the only landmark that helps you know where you are with the tracks. So, and this is kind of important to know that, for example, when you have deformation of the cord, it's quite hard to predict where the exact tracks will move because we don't see them. So this probabilistic is on healthy individuals, so we cannot really predict where the track go with a compression or a spinal cord injury, for example.
Sandrine, we have a question online from Caroline Landell. So she says, thank you, Sandrine. Are there any tools developed or currently under development that would be helpful to evaluate gray matter shape, for example, comparing contours?
No, there is not for now, but that could be something that's interesting. I think here there is like a whole field about really shape analysis that is quite far from what just simple metrics that we do here. But of course, it would require a bit more complicated things, but that could be something to look into.
I have another question down here.
Thank you for that talk. So I wanted to ask about spinal cord injury specifically. When you have cases where there is essentially a transection or a complete loss or disruption in the spinal canal itself. What is the margin of error? How gracious would you be when it comes to the segmentation, registration to the template, and core registration with diffusion data?
It really depends on, well, what you see in your image. I know sometimes it can be also like some metal implants and things that would just, if you have no signal, we cannot do anything with that. Of course, like in our segmentation model, we included data with traumatic spinal cord injury, for example, and we have some ground truths that were segmented from experts. So like those are things that we really tried that our segmentation model works and our canal segmentation works. Same thing for lesions because those are super important application. But if there's something that you cannot see with your own eye, our model cannot invent it. Like it's not better than your eye, it's just faster and less manual intervention.
Thank you.
Yeah, and I would say we, in some of our traumatic spinal cord injury work, we do try to warp the damaged cord to the template, and we're forcing it to do that. It does show some correlations with sensory and motor scores, but it's not perfect for sure.
Thank you so much for your talk. I have a question about the new features. I saw that there's a Furman score there in one of the images. How do you quantify that? Because it's a semi-quantitative scoring system. Are you doing clinical evaluation with radiologists? And how do you set your thresholds? I guess. Slightly hard.
Thank you. Thank you for the question. So this is work in progress. Like the validation part is a work in progress. So it is indeed a semi-quantitative metric that, for like you probably know it, but just for context for everyone in the room, this is using the information of the thickness of the disc that could be extracted from the segmentation of the disc, and the signal intensity within the disc that could also be extracted from the segmentation and the image. So based on that, we run a a classifier based on a cloud, like a graph with all the dots that has like this index. So we quantify an index from the thickness and from the intensity. And so we have a cloud, and then basically the idea is to classify, is to add create some threshold to basically classify those different cases into 7. Now the question is, how do we set the threshold? We set the threshold based on the spider data set, which is open, and I don't know if the spider has the Furman or if it's another. But we got some scores either from an open data set or from clinicians that we used to train a classifier. And then the step we're doing now is to have different radiologists review, basically assign a Furman score based on a data set, and then we're going to validate and potentially iteratively train the model based on that. If you're interested to collaborate on that, I would love it, because it does require multiple eyes to get something robust and valid.
A radiologist, but I do have some spine data that a radiologist scored for us. Okay. But it's like only maybe 70, 80. Okay. So I was really interested in comparing our scores to your automatic scores, because in the knee field, there's like no it's not a good it's not very reliable.
Indeed.
Yeah, so I was really interested in how you can get it reliable.
That's fun. So please send me an email, and then we can.
Yeah, I think a data set of 70 to 80 scored images is pretty good. Yeah, that's still big. Coming down this way. No, you're good. Okay.
So my question is about binary versus non-binary segmentation. So the examples you showed are binary segmentations, and probably for the spinal cord it doesn't make much of a difference to have a non-binary, but the smaller the lesion, for example, the smaller the structure, like a lesion, then it makes a difference whether you include the voxel or not.
Yes, exactly. So if we look, for example, at the MS lesion segmentation model, I'll bring back. You actually have the option to get the soft version. To get this kind of probabilistic value of the model, but even for spinal cord, it can still make some difference because of the partial volume at the boundary. But of course, like a T2 weighted have quite sharp boundary, and it's larger than the lesions. But yes, for like small objects, it's I think it's something we want to move forward to soft. But the problem is, we cannot really get like ground truth of this kind of uncertainty or of the partial volume. So that's the kind of thing that makes it harder. But I think now our MS lesion model, you can also have the soft version to do, for example, compute morphometrics with, because you can compute CSA with that, for example.
I see. And do you think it makes sense to sometimes to upsample, if you only have binary segmentation, to upsample the image so that you still do a binary segmentation, but you have a higher resolution?
I don't think, I mean, it depends. Then that wouldn't necessarily make that much of a difference because of the partial volume would still kind of be the same. You're just linearly and interpolating. Then it depends. Yeah, you're segmenting on the new one. So yes, your boundary will look nicer. But we have done some testing, for example, on spinal cord segmentation metrics, and we downsampled our image, and we tested what was like the variability of CSA was not that important. So I don't think it's necessarily like the perfect approach.
Okay, thanks.
More online. All right. Any more questions from the audience? Yeah. No. This is great.
I'm sorry. Yeah, just one more question. At the start of your talk, you were saying about how you were using the UK Biobank to look at with the T1 image what information or how you could segment the spinal cord. So I was wondering, for like just a standard T1 weighted image of the brain where there's like that little bit of spinal cord down there, what information can you get out of it?
So actually, this is I was measuring C2, C3 CSA. So I could get automatically the disc levels from here, and then I would measure, for example, CSA average on the upper cord. I was working with a subsample of like 1,000 image from the UK Biobank, and I had to exclude like 200 of them because there was not the coverage. So sometimes like it just depends the height of the person sometimes, or the image quality at the lower field was sometimes bad. But I think in the final study in this, I was able to measure it on 800 people. So that's also cool that you can do it on brain images, but usually just upper cord cross-sectional area that you.
So you don't necessarily need like if you're just looking at those upper areas and the field of view included the neck, you wouldn't necessarily need like a spinal cord specific acquisition sequence? Or?
No, this is this cool. Was really like a brain sequence.
Cool, thank you.
All right, any additional questions? All right, well, thank you, Sandrine. That was awesome. We've got the record for most questions, so let's see if that can be beaten tomorrow after the social and everybody knows each other.